Students of 10th standard, today we are going to end the chapter on the cell division, and we'll switch over to the structure of the chromosome and the study of the chromosome and the different structures of the base pairs and all these things, not in details but in as per required for the 10th standard. Now we'll begin with crossing over. Crossing over is a phenomenon which takes place in the prophase 1 of meiosis, and in the prophase 1 is a very prolonged phase, like it has the leptotene, zygotene, packitine and diploidine. Now in the zygotene phase, what happens, the homologous chromosome, they come near each other, we call it a dyad stage. So here we show with two different colors, one yellow and one purple color, to show the two chromosomes that in every person's cell the chromosomes are inherited from either of the parents. So this is shown in two colors. We are doing with one pair of chromosomes. Now in the packaging stage, each of the chromosomes, they break up into two sister chromatic. We call this two as the sister chromatic of the homologous chromosome. Now in the diploidine stage, the sister chromatic of the homologous chromosome, some of the places they come and entangle or join together in certain points, in one point or in two or in multiple. Now these places are called the chiasmata. Now in this chiasmata, what happens, there is a genetic material exchange. You can see the yellow part here has, the purple part has crossed this type and yellow has got this side. So exchange of the gene takes place. Now these genes are basically all the similar type of bearer of characters. That means they exchange the genetic materials of the same character. Now when they separate at the end of the prophase 1, now you can see here that some of the chromosome or some of the gene has exchanged. Now this is what we call crossing over, and crossing over leads to variation. So variation occurs in every offspring. You can see the gametes which are carried, they are either recombinants or through random fertilization and an exchange of the chromosome. All this leads to variation in the offspring. They are different from the parents. There are some characteristics you will see that in the offspring they are similar to that of the parents. Some of the characters are there which may be dissimilar, which may be little bit different from that. So those variations, one of the reason is crossing over. Now we will study a little bit about mutation in 10th standard. Now what is a mutation? Mutation is there are certain change in the structure or the number of the chromosomes. Now this change in the structure of the number of the chromosome can arise due to various reasons. It can be due to certain physical factors, due to some chemical factors. So you will see that these are called the mutations. Now these mutations are mostly heritable. They can be very slight, they can be large. Usually mutations are not so useful. They can be harmful, they can be lethal, some are deleterious. So and maybe in the natural mutations are useful. Many of the things that we see, many of the vegetables that we see are product of natural mutations. So they are good, but there are artificial or induced mutations are also there. Artificial and induced mutations are done with help of chemicals, with the help of chemical mutagens like 5-bromouracil. Even there are various chemicals which are also which they induce mutation. Ultraviolet rays are mutagen. Cosmic rays, or say that the gamma rays are mutagens. X-rays are mutagens. So you can induce mutation to get certain effect. So many of the things are created, many genetic materials are changed in certain organisms through induced mutation. So what is a mutagen? The factors which initiate mutation is called a mutagen. So there are types of physical mutagens, like you can give an example of physical mutagens, say ultraviolet rays or say X-rays or gamma rays. Chemical mutagens are also there, like you can say benzene is a mutagen, 5-bromouracil is a mutagen, and this type of substances which can bring change in the chromosomal structure, change in the base pair by addition or deletion or insertion of the base pair can lead to various types of mutations. Now that much is what we require for 10th standard. You have to know all this about mutation. Now we will go to the study of the structure of the chromosome. Now chromosome word comes from the word chroma, which means colored, and soma means body. So these are colored bodies or structures which are found inside the nucleus. Now these are the bearer of the heredity. They are highly coiled structures, sometimes seen to be when you see under a microscope, you can see there are alternate light and dark bands. The light bands are called the euchromatin areas and the dark bands are called the heterochromatin areas. And there are certain places, this region is called the primary constriction and this particular structure is called the centromere. Now this is also a single chromosome which is single chromatid here. But sometimes this chromatid, they break up into sister chromatid, and you can see this is also a single chromosome but having two sister chromatid. So if this is the primary constriction, some chromosomes like 11, 12, 13, 14, 15 number of chromosomes of human genome, human chromosomes, they have a secondary constriction also. Now this is called the secondary constriction, and beyond the secondary constriction there are certain projections which are called the SAT chromosomes. So these chromosomes are highly coiled structure and they bear the gene. So what is a gene? A segment of the chromosome that carries the hereditary material is called the gene. Now these genes are expressed in form of certain types of nitrogenous base pair which we are going to study in the due course. So what do the genes bear? The genes bear certain characters which are expressed in two or more alternative forms, and these are called alleles, which you have already studied. Now let us go and discuss a little bit more about the structure. So when you break up this chromosome, basically suppose this is a chromosome. So if this chromatid is seen, they are coiled around, there is a supercoiled structure like this, and they are coiled around 8 molecules of protein, particularly in the eukaryotic cell. These proteins are called the histone proteins. Histones are found only in eukaryotic cell, they are not found in the prokaryotic cell. And in the prokaryotic cell, that's why it's called a genophore. So this coiled structure is actually, it's a supercoiled structure that makes the condensed chromosome look like this. But the chromosome is not always as thick. Sometimes the chromosomes are also found in form of fine thread-like structures like this and beaded structure. So these beaded structures, these are called the chromomeres, and these fine thread-like structures are called chromonemata. So like in prophase, early prophase, they look thread-like structures, but if you see in the metaphase stage, they will appear thick like this. So when you see this fine thread-like structure, this thread-like structure is actually making up the double helix, what we say the Watson-Crick model of the DNA. So the double helix looks like this, you can see here. This is the DNA double helix, okay, and this is the axis, or what we call, they are joined together, and here the base pairs are joined. Now what it is made up of, we are going to study in the due course. Let us see. So you can see the double helical structure. So this is actually the chemical structure of the double helix. So we say it's a double stranded polynucleotide chain. So actually here, this is the nucleotide. What is the nucleotide made up of? This is made up of certain purine bases, a sugar and a phosphate group. So what is a nucleotide? We are going to know that a nucleotide consists of, so a nucleotide consists of a sugar, a phosphate and a base. So a phosphate, which is a phosphoric acid. This is a sugar which is called a pentose sugar because it has 5 carbon. We call it also a ribose sugar or a deoxyribose sugar. If it is ribose sugar, then it is C5H10O5. If it is deoxyribose, then it is C5H10O4. 1 oxygen less, so we call it deoxyribose. So in DNA, it is a deoxyribose sugar, so that's why it will be C5H10O4. So it has 5 carbon, this is the phosphoric acid, and this is the base pair. What are those base pair? We are going to know there are 4 types of base pair in DNA. adenine, thymine, cytosine, guanine and cytosine. Now among this, the adenine and the guanine, these two bases are called the purine bases, and thymine and cytosine, these are called the pyrimidine bases. So we say, what is a nucleoside? If it is a nucleoside, we say a nucleotide, a phosphoric acid, a sugar and a nitrogen base is called a nucleotide. What is a nucleoside? If you remove this phosphoric acid, only the sugar and the base will be called the nucleoside. So how it will be the nucleoside named, or how it will be the nucleotide named? Suppose the sugar is adenine, the nitrogenous base is adenine and the sugar is there, then we call the nucleoside name will be adenosine. And if we add the phosphoric acid to it, the inorganic phosphate which is from the phosphoric acid then we say it is adenosine or adenylic acid. So adenylic acid will be a nucleotide, and adenosine, this is called the nucleoside. So you have to know the difference between a nucleoside and a nucleotide. We are going to study in details about the structure of the DNA in the next class. Till then, be safe, and if you have any questions, you can put up the questions, and we are going to discuss in the due course. Thank you very much.